My project is how do endophytic bacteria from mango affect the growth of the plant pathogenic fungi Botrytis cinerea and the use of 16S rDNA sequencing to identify bacteria. So endophytic bacteria are bacteria that live inside the plant and they benefit both themselves and the plant host. Botrytis cinerea causes stem end rot and gray mold disease in mango plants, and I wanted to test to see if endophytic bacteria from mango could play any part in inhibiting that growth and be used as a biocontrol agent. So my project is a continuation from last year where I tested endophytic bacteria from mango and its effect on the fungus called Totricum gliosporioides. So basically what I did is I isolated the endophytic bacteria from mango. I cut the stems of mango, I ground those samples and surface sterilized them, and I ground them in a saline solution. I planted the plant suspension onto tryptic soy agar media plates, and I grew the bacteria and incubated them at 30 degrees Celsius for 24 hours. After that, I placed a fungal disc of Botrytis cinerea and I placed it onto the plate, and I let that incubate for 24 hours as well, and I observed the growth from that. And down here is my data from measuring the diameter of that fungal colony, and there weren't many physical visible results. However, my statistical analysis shows that there was a significant difference between the control and the tested samples that I had. So the next part of my project that I did was trying to further identify this bacteria using the 16S rDNA sequencing. So I ran a PCR gel electrophoresis, and I performed the gene clean, ligation, heat shock transformation, and plasmid isolation. And then finally I ran the PCR just to check to see if my inserts were there. And these pictures down here show the bands, the results from the gel electrophoresis, and after the heat shock transformation with the plasmid isolation, those two colonies there, so the white colonies were the colonies that showed my the bacteria with the insert in it. And yeah, so in conclusion, all bacterial colonies were observed. My next step right now is to further send my data to a company to further sequence and send back the base pairs for my bacteria, and once I get that back, I'll be able to compare it to a database and hopefully be able to identify what this bacteria is. And like I mentioned earlier, the bacteria did have an effect in inhibiting the growth of Botrytis cinerea, and hopefully in the future more studies will be done and I can do more research into see how endophytic bacteria can be used as a biocontrol agent.
My project is called Building Bridges, where I tested 3 different bridge designs to see which one had the highest strength to weight ratio. I basically had the 3 different ones, and I weighed each of the bridges before I broke them, and then I put enough weights on them until they collapsed to find which one could hold the most weight in comparison to its own. This one, the arch bridge, ended up being able to have the highest strength to weight ratio, but the truss bridge could hold more weight overall. This one was just more effective in using its own weight to hold up more.
My project here today is about how different salt concentrations affect the germination of radish seeds. So what I had hypothesized that was 25 grams of salt diluted in 50 milliliters of water would be the best concentration for the plant growth. And what I found in my studies is that that was the correct hypothesis and that I was right. So the other concentrations I used were 0.5 grams of salt in 50 milliliters of water, 0.75 grams of salt in 50 milliliters of water, and 1 gram of salt in 50 milliliters of water. And then I had my control with no salt. Here on my different data tables and my graphs, you can see that the 25 grams of salt was significantly higher than any other of the concentrations, and that it helped the embryonic root length of those radish seeds be longer than the rest. So that is my project.
So in my project, I wanted to test how we could use algae in order to reduce the amount of greenhouse gas emissions in our atmosphere. And I did this by testing how we could use algae and whether it needs to be in the presence of sunlight or without sunlight in order to remove the amount of CO2 levels. And I did this, my hypothesis was that that the test tube that had algae in it and was exposed to sunlight will be able to remove the amount of CO2 levels and return the solution back to its original blue color. My materials were bromethadine blue solution, which is a pH indicator. I also used a straw, 4 test tubes, a test tube rack, 4 stoppers, aluminum foil, a light source, and a marker and a tape. So my procedures were I first poured 80 milliliters of water into a flask, and then I put 8 drops of bromethadine blue solution into the water. Bromethadine blue solution is a pH indicator, and it gives off a blue hue. After the pH was into the water, I then, with the straw, gently exhaled into the solution, which the carbonic acid from my breath made the blue solution turn yellow. And after the solution was yellow, I then poured 20 milliliters of the water into 4 of the test tubes. And then I got green filamentous algae, and I cut into 2 equal pieces. I then put the 2 pieces in 2 of the test tubes. I then put a stopper on all of them and marked them 1 through 4. After they were marked, I put aluminum foil in one of the test tubes that had algae and one of the test tubes that did not have algae. As you can see here, this is the test tube that had the algae, and this is the test tube that did not have the algae. I put the test tube under a light source and overnight, and then the next day I came back to record the results to see any of the color changes, and my hypothesis was correct. The algae that was exposed to sunlight was able to remove the amount of CO2 caused from my breath and return to its original blue color. And this is all important because greenhouse gas emissions in our environment have a lot of negative effects, such as health issues, air pollution, global warming, and so on. So we need to find ways on how we could reduce the amount of CO2 levels in our environment. Actually, 76% of our greenhouse gas emission is carbon dioxide. And through my research, I found out that algae is one of the most effective ways in reducing the amount of carbon dioxide in the atmosphere. Some algae are actually 10 to 50 times more effective in reducing the amount of carbon dioxide than other terrestrial plants and trees. So through all my research, I found out that the production of algae farms are one of the most effective ways in reducing the amount of greenhouse gas emissions in our environment to make our Earth a cleaner place.
Our project is Does Anybody Really Know What Time It Is? That is a reference to the Chicago song, and the question that we asked in the experiment was we were trying to find the difference in time between Greenwich, England, which is where the prime meridian runs through, and St. Lucie County, Florida, using the different measurements of time. And our hypothesis was that standard time would say around the same mark for the entire experiment because of the way it's set up to do better coordination between the different parts of the world. And we thought that solar time would remain mostly constant because of solar days being 24 hours in length. And we thought that sidereal time would largely trend one way because sidereal time has shorter days, being 23 hours, 56 minutes, and 4 seconds.
How we got our results was going from Greenwich, England to St. Lucie County and calculating the difference in minutes in solar, sidereal, and standard timing to find the results that we did, which ended up being that standard timing had remained the same. Solar timing had fluctuated a bit more than we expected, and sidereal time had fluctuated a little bit less than we had expected.
So basically, it proved one of the three statements in our hypothesis was proven correct, that being the one about standard time. As she said, solar fluctuated more than we expected, sidereal fluctuated less than we expected. And I believe that's everything.
So my science project, I put my question as, what holds two boats together better, a screw or a nail? and I think this can help other people by showing them what to use when they are building things, because whenever you're building things, a big part of it is finding out what to fasten your stuff with, and if you don't fasten it with the right materials, it could break and fall apart while you're using it. I mainly chose this experiment because I have a lot of background information on this experiment, and I work almost every week building stuff and working on cars and stuff, so I can relate to this. So whenever I started this experiment, I made the hypothesis of the screw will hold them together better because it has threads, which puts a grip and traction in whenever it has force on it. And I thought that because whenever you have two things and you have force on it, it tries to get ripped out, and whenever you have threads, it doesn't want to get ripped out. Whenever you have a flat surface like a nail, it just pulls right out. After I did the experiment, I came to the conclusion that the screws did work better, because as you can see right here, after I stood on it, it instantly ripped out, and after I stood on the one with the screws for about 5, maybe 6 minutes, nothing happened to it, and it stayed intact. So whenever I was doing this, I started by gathering all my materials, and I cut the wood into equal planks. I put the two screws into the side of the screw one and 2 nails into the side of the nail one, and then I carried on with the experiment.
I think that through the years my projects have kind of geared more towards my interests. When I started when I was younger, I was looking more at things in the technology area, and as I've grown up at Treasure Coast right now, we have a biotechnology program, and one of the things that I'm really passionate about is biotechnology. So being able to expand upon the fields of research that I'm passionate about has been a great experience. One of the inspirations for my project was actually my mother. She has a condition in her shoulder right now, and some of her cells are currently having some issues regenerating. So I was looking at things that can kind of slow cellular aging and stop the clock in a sense. It was really interesting. I learned a lot of things. One of my judges was actually specializing in the field of antioxidants, so it was really fun to hear more about his perspective and some of the things that I could improve on my experiment. I learned a lot, definitely.
This is my project, and I called it Daphna. So basically, Daphna is an indicator species, which means that they can detect abnormalities in their environment. Basically, that if their environment is being degraded, then they can detect that. So a lot of the times, if their environment is being degraded, Daphna either dies off, and that's an indication that something needs to change, and especially something in the groundwater, because a lot of pollution can get into groundwater from pesticides and stuff like that. So my project, I established a stable Daphna culture, and I took different samples from a parking lot of sediment, because parking lots, obviously, they may contain a lot of pesticides, stuff like that. So I took those samples, and I took different amounts of sediment and put 250 milliliters of spring water into different containers. So the highest one I took was 4.62, and then 3.02, 2.06, and then I had one with no sediment at all. So I put 40 Daphna in each of those containers to see which container warranted the most amount of deaths, stuff like that. And actually, my results came out to be that the sample with no sediment in that at all had the highest deaths. So maybe more trials need to be performed and stuff like that to see, like, to actually determine in the future. So that's what I plan to do for my continuation of this project next year.
I guess the main intention of our project was to find what shape or size parachute would work best for slowing the descent of an object's fall. And when we were experimenting, we found that a square parachute with lips over the side worked best at catching air and slowing the fall. This is the bottle rocket that we use. We had put this into the bottom, and with a pump, we had pumped air into it until it had built up enough pressure to shoot up, and then once it shot up, we had this thing come off and release the parachute, and it would catch the air, and we would time how long it would take from the apex until it hit the ground, and we recorded so we would know exactly when it hit that apex.
And my project title is Enlisting Artificial Intelligence in the Service of Restoring the American Dream. This project aims to create a tool using artificial intelligence that policymakers can use to predict the outcomes of policy decisions in low-income neighborhoods. Research has shown that strong correlates to upward mobility in childhood have a very big impact on outcomes in adulthood. So using this project and the machine learning tool created from it, we'll be able to quantify the impact of policy initiatives to improve the place effects of specific low opportunity neighborhoods. The cost of implementing said policy will result in net gains through taxes accrued from the resultant higher income bracket. Widening income inequality is the defining challenge of our time. In advanced economies, the gap between the rich and poor is only widening. Analysis from the International Monetary Fund has shown that the income share of the top 20%, when it increases, the GDP decreases. But when the income share of the bottom 20% increases, the GDP also increases. So I used TensorFlow to create the machine learning algorithm for this project, and I focused on a research paper from Harvard researcher Raj Shetty, who is nominated for a Nobel Prize in economics. My results found that the purpose of the AI would be used to find the specific correlates to mobility so that policymakers can manipulate them using the web tool to find the actual outcomes in very localized areas of their policy.
This is my project on what's the fastest way to cool a soda, which I would say I chose this project because some people may like their drinks cold, so I chose it, and then I tested it out of the 4 cooling ways. I tested the refrigerator, freezer, ice water bath, and an ice only bath. And my hypothesis was honestly that the freezer was going to be cooling at the fastest rate, but turns out ice water is the fastest way to cool your drinks. And so my hypothesis was proven wrong by my data as seen here. And from my data analysis, I was pretty surprised seeing how ice water started at 20.33 degrees Celsius and it ended with 2.35 degrees Celsius because it was a decrease of 17.8 degrees Celsius just in 30 minutes. The cooling devices is what I controlled, was my independent variable. I had the freezer, refrigerator, the ice only bath, the ice water bath, but then like what I measured was the temperature of the sodas, and my constant variable was the cans of Coke Zero.
And this is the potato battery project. And we did like an open circuit with the potatoes. And we did like different like types of potatoes, like regular potatoes, sweet potatoes, and like red potatoes. And we like measured the voltage of them to see like how much power they could give.
We also boiled the potatoes and put a lemon and a banana to also test how much voltage energy they have, and to compare the two to see which one had more. The boiling the potatoes didn't do anything at all, but which is kind of sad, but it was still a good try to test it. We also weighed them to get them remotely similar, so we know the control variable. And the variable that had the most voltage energy was the lemon.
Yeah, we also wanted to try like fruits and stuff, but we couldn't get any more because it was like a last minute project. So like we did a lemon and a banana, and the fruits had like the most voltage out of all of them. But we didn't boil the fruits because I feel like that would like make them, you know, like melt or something like that.
What I really loved about it is because there's so many companies that, like with mosquito repellents, they use a chemical called DEET, which has also been like, though yes, it's really effective and is proven to be probably one of the best, it can also affect us as humans as well. So what I like because usually my community has like a lot of like mosquitoes all over, and same thing with my partner. So then we just like, I want to repel these mosquitoes, but I don't want these kind of chemicals. So we use natural oils instead, which have been proven to be effective. The experience today, I loved it. It's like connection between one and one person. It just like really inspired me. So it doesn't feel like a project. It just feels like a natural hobby. It just feels like interacting with multiple people, and that's something I love. Just like everybody comes together to do this one thing, you know?
Okay. And
this is my first time doing a science fair right now. So then what I plan in the future is just improving maybe on this, or maybe doing something completely new. But I like to go on the same basis because like this is like an experience. It's a whole learning experience, and that's what I also love about this too. Like just learning in general it's an amazing thing. It's a beautiful thing. Knowledge is probably one of the most powerful things you can have. So then that's why I love this. I want to like improve on this or just start on something new. I would like to like maybe even do this exact same thing in the summer, because like I did this like when winter was coming, so less mosquitoes come in. So then like doing this in the summer will like be really good for data collection. And I also want to maybe even compare this against like actual repellents, because I just compared it with like natural repellents that I made and just see which one was better, just to see if it's even effective. Well, now maybe like next year, or like if I plan on doing it, I want to maybe compare it to other like actual brands, you know?
Today our project is on which toothpaste works the best. So we wanted to know which toothpaste works the best, and for this we performed a Kirby-Bauer test, and we used 3 types of toothpaste that had 3 types of chemicals in it. One of them was hydrogen peroxide, the other one was fluoride, and the other was triglycerin. And we hypothesized that the toothpaste with triglycerin would work the best. And after we performed our Kirby-Bauer test, well, before, we actually had to swab her mouth for the bacteria, and we had placed them on nutrient agar plates. And our 4 variables was water, hydrogen peroxide, fluoride, and triglycerin. And we swabbed her mouth and we put them on the plates, and then we left them in the incubator for around 3 days, right? Around 3 days. And once we came back, we had our results. And based off of our results here, hydrogen peroxide had the greatest and largest ZOI, or the zone of inhibition. And based off of the zone of inhibition, it tells you which chemical had inhibited the most bacteria, and since hydrogen peroxide inhibited the most bacteria, it did work the best compared to triglycerin. So our hypothesis was actually refuted, and hydrogen peroxide did work best compared to triglycerin. We chose triglycerin based on the reviews. We saw that the toothpaste with triglycerin, there was a lot of people saying how good it worked and how great it worked, so we just initially thought that that toothpaste was going to work better.
So my project is about whether or not oregano oil can be used as an antimicrobial agent for household services. So what we did, we basically used Q-tips and we swabbed them around like 3 different locations of the household, like the doorknob, the AC vent, and my iPad. And we used like little zigzag patterns on agar plates, which is filled with nutrient broth, and we closed them tightly and we put them in a homemade incubator, which was set at a temperature at approximately 37 degrees Celsius. And after a few days, we did the treatment, which was paper discs soaked in 2 to 3 drops of oregano oil, and we placed them carefully with tweezers, like in a square shaped, and as you can see, there's like little reduction around the AC vent compared to the iPad cultures, compared to the garage. So, and I wanted to do this project because oregano has 2 bioactive ingredients that have shown to have anti-cancer properties and food preservatives. So I wanted to see if essential oils can be used as household services for antimicrobial agents.
And this is our project Breaking Point. So what we did, we got rubber bands and we placed them in different temperatures of water to see how the effect of the temperature would on how far they stretched.
What we found was that the colder temperatures actually made it stretch farther than normal temperatures, but the warm temperatures, the hot one, made it stretch the farthest and before it broke.
Yeah, so the data, it does support our hypothesis, but there were some outliers in the process, and it overall did support the hypothesis. Yeah. We use the same type of rubber band with different temperatures of water to keep less variables.
Well, my project is about germs in school. So many people would ask themselves, where's the germiest place in school and where's the dirty place? My hypothesis was that it was the bathroom. But after doing some research and my presentation, I figured that it was the window. I found out that it was the window, and that was really interesting to find out that it was a window that was the most germiest place. Because when you think about it, I would think it would be a desk or a bathroom or anything but a window. But when I swabbed it, the most place that had germs was actually the window. It first started with the little spots, and then a lot of mold and smell came out of it. The most smelliest one, I would have to say, is the school desk. It was really smelly and nasty, but my hypothesis wasn't really wrong, because a hypothesis is really an educated guess. It was just a little off because it was the window. So I really enjoyed doing this presentation and project, but if I would have to tell somebody to do it, you would have to have a lot of time and patience to do this. If not, then it's not the right project for you.
My project is, does the type of yeast affect how much bread rises? I came up with this project because I like cooking, so I thought it would be interesting to incorporate cooking with chemistry. And thus where I came up with the idea, does the type of yeast affect how much bread rises? The 3 yeasts I used were instant, rapid rise, and active dry yeast. My hypothesis stated that if yeast affect how much bread rises, then I believe that the rapid rise yeast will make the bread rise the most, because it allows the yeast organisms to eat the sugar and starches in the bread, which produces carbon dioxide and causes bubbles to form and allow the bread to rise. And after I had finished making all the breads for each of the trials, my hypothesis was in fact correct. I had measured them by measuring the height once taken out of the oven and the height after cooling down. And then, because once the bread has cooled down, it does deflate a little. So I had added up the measurement for both the trials for each, where I got 18.9 for trial 1 and 16.3 for trial 2. And in total, I got 35.2 centimeters. And I continued doing that for each of the trials until I got the total for each one, which revealed that rapid rise yeast did rise the most. Like I had stated in my hypothesis, it would.
Social media, like everyone today.
I'm just amazed with the presenters. Middle school is just, I've learned so much. Normally as a parent, we're so used to teaching, but I felt like a student today. Very well done.
I have to agree with Dorothea, Miss Oates here. I've been judging the science fair for probably the last 5 years, and I feel like what I've seen today has been a much better quality than what I've seen in the past, that the presenters were really, the students were really well prepared to present and speak to an adult with really great interviewing skills. So a lot of real world application in what I saw today, and very impressed with what I'm seeing.
As a retired science teacher from St. Lucie County and being at this event every year for the past 10 years, I am really amazed, especially with the COVID and everything that's going on for these kids to put the maximum effort through in these projects. And it really, really says a lot about our county and the wonderful job our students are doing. Bravo to all the students here.
My project is a solution for noise pollution. So noise pollution is a growing issue in cities, and even smaller ones, because the population's growing, the amount of traffic is growing, and overall there's just more noise. Even at night, like in the early morning, late hours, it's a big issue. So the solution for this that I've found is sound foam, also known as sound panels. And you put these up on your wall, and they're in the shape of wedges, and it practically just gets the sound waves and cuts them in half, and it cuts down on the amplitude, which is like one of the biggest issues why sound is so bad. And my hypothesis was if I use sound foam, the noise will be reduced by at least 10 decibels because the foam reduces the amplitude of the sound waves and dissipates sound energy. And my hypothesis was true. I found that there was a high of 104 decibels on my control, and I was measuring the highest decibels for a span of 5 minutes. So I was testing for 5 minutes, the same traffic noise, and 104 was my high. The box without foam was 92 decibels high, and the box with foam was 79 decibels. So it was over 20 decibels cut down on, which 104 is actually really bad for your hearing, and 79 is fair range, so that's good. And I could have chose different foam, like there's higher quality foams of course, but for this project I was going based on like what's the cheapest solution, what's the most accessible, and it worked.
And for my project, I decided to try to determine the function of a protein by using the amino acid sequence and the mRNA sequence. So like over evolution, the genes of certain animals, they mutate. So the genes will change, and as they change, the animals become more diverse. And the sequences of the genes, some of them align with the common domain, so they can remain the same protein. And in order to remain the same protein, they have to use the same domain. And to investigate that, I decided to look at the sequences of protein and mRNA and keratin. And so I decided to use keratin because it was something I was more familiar with. And so I used the NCBI database so I can look at the protein and mRNA sequence of the human species. So I compare it to other species and find similar sequences in the bat, chimpanzee, and also the cat. And so as you can see here, these are the sequences of the keratin protein and then in mRNA for keratin. And looking at the similarities between the sequences of all the organisms, there's like different ranges of how similar they are, so I can get a diverse results for looking at the sequences. And after I compared all the sequences, I found more similarities in the protein than in mRNA, which is probably because the protein sequences remain the same, so they can remain the same function. The mRNA sequences are more diverse since each species have their unique DNA, so they're gonna be coded for different things. And as analysis, I saw that the proteins shared more similarities in the mRNA sequences. And also, the main thing of domain was the intermediate filament domain, which is responsible for like cytoskeletal, so it's like structure in the cells. So I saw that the multiple sequence alignment, which I saw, showed that the intermediate filament domain was responsible for the function of the keratin, which is structure in like your hair, your nails, and stuff like that. And then for application, a lot of the stuff that I researched was comparing the sequences. They can be used for 3D analysis of the domains, and as well as looking at the evolution of the certain sequences, so you can compare each sequence between different organisms or like different bacteria as well. So you can like potentially solve diseases and prevent them from happening by looking at the proteins that cause them.
My project is, are your foods genetically modified? So why I did this project is because I wanted to find out if my foods that I was eating every day are genetically modified, because I do consider myself to be quite a healthy person. I care what I fuel my body with. And as of 2022, there's a new law that you have to, companies have to label their foods as bioengineered or not. So my hypothesis was that if I do use these PCR techniques in the gel electrophoresis, that I would find that that there was some genetically modified genes in it. So what I first did was I used 2 controls, positive and negative controls for genetically modified genes, and I used my 4 test food samples. I used corn muffin mix, white cornstarch, tortilla chips, and cheese puffs. I use these because of the corn that they do have in it, since this is highly reliable for genetically modified genes. I did also use these because just I like them and I wanted to see if there were any genetically modified genes in them. So the first thing you do is you find a slurry with these 4 things with just distilled water. After that, you mix it with a primer, which then you are ready to do your PCR test, which the PCR test is what you use for COVID testing too, so I thought that was really interesting. It's very tedious. You have to use a lot of master mixes, a lot of primers. I used a different plant primer and a GMO primer to see if that maybe had some effect on my experience. And after I did all of those master mixes for my controls and my 4 test foods, I was ready to do my PCR testing using this right here. This took a lot of tries to do and a lot of trial and error. But we figured it out, and after PCR testing, you put these in these gel, and these gels are pre-made, and I put each of my samples in these 7, so my controls, my 4 test foods, and then my 2 positive controls. And then I run these through a gel electrophoresis, and it's basically attached to a positive and a negative, and it should move and create bands, as you can see. So my conclusion is that my project was inconclusive because the bands, as you can see, they aren't very apparent on here. There could be a lot of reasons why this could have happened. First reason, I could have chosen maybe the wrong food samples. Maybe if I had gone for raw foods, then maybe that could have been better and my experience would have worked better.
if I would have made my own gels, maybe that could have been better. And also the kit that I had used from Biorad, it says clearly that you have to store it at -20°C, and since we do live in Florida, maybe it got denatured because of the heat. That could happen in the classroom just by transportation, which things that I could not have controlled. So just a lot of problems may have occurred, yes. But overall, I have learned a lot from this experience, just about how much genetically modified genes are in your foods, and just some people in general don't even know that there is DNA in food. And when I would tell my friends about it, they're like, "There's DNA in food." Yes, there is, and so there's a huge debate on if it's scary or not. And that was basically what my project was about.
And my project is called Fallen Arches, the Surprising Strength of Eggshells. In this project, I'm testing out how much weight the arch of an eggshell can bear. At first in the project, I thought that the weight wasn't going to be a lot, and I thought the eggshells were going to break really easily because a lot of people tend to think that eggshells are fragile and they can't hold a lot of weight. But as I was doing the project, I realized that there was a lot of weight that was being able to hold on top of it. There was a lot of weight that the eggs could hold, and I did multiple trials to test it out to see if my results were true every time. And as you can see, there was a lot of magazines being put on top of the eggs, and there was a lot of weight that the eggshells could hold overall.
Basically, my board is on the effectiveness of Murad and Survey, the most popular facial cleansers for right now in the industry. And I did the effectiveness on how well they can clear out bacteria that cause acne. And so basically what I did the Curry-Bauer disc method, which allows to see what's like the bacteria around it, what stops the disc. So basically, I placed 2 sterile discs with the medication on, and then I placed it in with the lawn streak method culture. I placed the disc on top of the E. coli, and I put it in incubation for about 2 days to see and allow for the E. coli to grow and proliferate. And I went to go see how much it stopped the growth of the bacteria. And basically, my method came out to be it didn't stop any of the growth. So what came out is that the acne, the facial cleansers, Murad and Survey, are not effective in clearing out acne as much people say it does. And so that my hypothesis was Survey would clear out the stop the acne growth more or the bacteria growth more, and it did not. They all had ineffective results.
My project is on how do different chemical and physical reactions affect the launch height and the max height achieved by a plastic rocket. So the 4 different chemical reactions that I used were Coke and Mentos. I used vinegar and baking soda, Alka-Seltzer and water, and elephant toothpaste. And I just wanted to see how those different types of reactions would affect that rocket. So the Coke and Mentos reaction is a physical reaction, and it has to physically interact with the Coke in order to produce carbon dioxide, which builds up in the rocket and causes it to launch. The vinegar and baking soda is just a normal reaction with vinegar as an acid and baking soda as a base, and when they react, it produces carbon dioxide. And that causes an expansion in the rocket, which also causes the rocket to launch. Alka-Seltzer and water is a little bit different in that it's an aqueous solution, and the whole reaction happens when it comes into contact with water. And both the base and the acid are both compacted into an Alka-Seltzer tablet. And when it reacts with the water, it produces carbon dioxide, just like vinegar and baking soda. And elephant toothpaste is different because it has a multiple reactants to multiple products type of reaction, in which you have to use yeast and water and react them with hydrogen peroxide and soap, and they cause an oxygen expansion, and as well as a foam expansion. The oxygen is trapped inside the foam produced by the soap. And so I used those 4 reactions, and I did some research on them first, and then after I did my research, I made some procedures in order to conduct my reaction. And I did 2 trials of both of my reactions, and I recorded the heights and how long it took for the actual reaction to occur. And so my hypothesis originally was that the vinegar and baking soda would go the highest and the Coke and Mentos would go the lowest, because Coke and Mentos is a physical reaction and vinegar and baking soda is a direct acid to base reaction. But my hypothesis was mostly correct, but some of it was not supported by my results, as Coke and Mentos actually did go the lowest. Vinegar and baking soda did go the highest, but the height to reaction time ratio was higher for Alka-Seltzer in trial 2, while I had predicted it was going to be higher for vinegar and baking soda in both trials. And there is some things that I realized I could have changed about my project, which was the Coke and Mentos and how much amount I can use in order to best conduct their reaction. And also the elephant toothpaste, if I could use stronger reactants, which would fasten up the reaction in order to produce even faster oxygen expansion, which would cause the rocket to launch even faster. And my reason for doing this project was mainly just to figure out different types of reactions, research them, experiment with them, and relate them to real-life rocket launches, as they use combustion reactions to launch real-life rockets, and see if they could just explore in the area of experiments and reactions to see which type of reactions would better launch real-life rockets, and how we can find more ways and more reactions that would better launch those rockets, and we would find a new era of space exploration if we could manage to do that.
Here is our project. So our question: which paper airplane will travel a further distance out of these three models? The world record fastest paper airplane, the F-15, and a classic paper airplane.
Our research was paper planes, just like real airplanes, use the same four aerodynamic forces that allow them to lift and stay airborne: thrust, lift, gravity, drag.
So our hypothesis: my partner and I believe that only the world record fastest paper airplane will fly a further distance than the classic model and the F-15 model.
The method we used was taking each individual piece of paper and fold them differently to see how the adjustments we add would affect the way the paper airplane would fly. The first type of plane was the classic plane. For our attempt 1, we got 27 feet. For our second attempt, we got 23 feet. For our third attempt, we got a distance of 14 feet and we straightened the nose. Our average distance was 21 feet. And for the second model, it was an F-15 airplane. For attempt 1, it was 27 feet. The second attempt was 33 feet. And for attempt 3, we got 18 feet and we straightened the nose and wing outwards. And our average distance was 26 feet.
And for our last trial, for our first was the design was the world record fastest paper airplane, and the first attempt it flew 28 feet. For our second attempt, it flew 25 feet, and for our last test, the world record fastest paper airplane flew an outstanding 70 feet, and we straightened the nose and wings. The average distance was 41 feet. And our conclusion: our hypothesis was correct. The world record paper airplane flew the furthest distance out of all three models we used. Making small adjustments to each single paper airplane and testing them 3 times each, we learned that each individual plane traveled different distances. The adjustment made onto the world record fastest paper airplane model was by straightening the nose of the plane and straightening the wings on both sides made the paper airplane travel a furthest distance from the other models. After analyzing the chart, shows that by making small modifications to the paper airplane helped maximize its distance. We believe that was the scientific method of our experiment.
So my project this year is on Stop the Clock, a radical idea. So when we look at cellular aging and things that cause chronic diseases such as cancer, heart disease, Parkinson's disease, we try to figure out what causes those diseases. So one of the chief agents in causing these things is actually free radicals. Free radicals are molecules that have unpaired electrons that can cause extra damage in your body. And free radicals are produced in simple things such as metabolism when you eat your food and it gets broken down, or things such as tobacco and radiation poisoning. So things that can prevent these free radicals from causing extra damage in your body is actually antioxidants. So antioxidants work to remove that oxidative stress from your cells and ensure that the free radicals don't cause any extra damage. So in my experiment, I looked to see how these antioxidants can slow the process of cellular aging. So I looked at a total of 5 different antioxidants. I looked at NAC, vitamin E, B12, vitamin C, and water as my control. So throughout my experiment, I noticed that NAC and vitamin C did the best at removing free radicals from the solution that I used with ramen noodles to kind of act as the muscles in your body. So through doing this experiment and finding out that NAC and vitamin C did the best at removing the oxidative stress from your body, it helps us to learn how we can implement new things into our diets and things into our everyday lifestyles that can improve on our living and slow the clock on cellular aging.